we're going to be talking about linking sequence variation in terms of single nucleotide variation all the way up to these large structural variants to chromatin dynamics and how topological data types can really help us understand what these variants are doing. So the outline of today's talk, we're going to do a brief introduction into the world of chromatin conformation, what the data type is, how it's generated, how it's visualized, and really kind of what that means for us in terms of bringing that data into understanding chromatin dynamics and gene regulation. Then we're going to move into a few case studies in the primary research area that do some really phenomenal jobs in terms of linking SNPs and GWAS data to their appropriate actually gene that they're impacting. And then we'll talk a little bit about structural variation and how that really changes the chromatin dynamics and how that actually influences how genes get regulated. And then finally, we'll spend a little bit of time talking about how you would integrate these 3D topology data types into what you're currently doing today, and maybe how we have a solution that might kind of encompass many of the different questions that you're already asking with sequence-based data, and you'll get an additional layer of that 3D information in it. So let's get started. The world of chromatin conformation. So I think to set the stage here, I want to kind of point out the fact that many of approaches to genomics really result in this linear view of the genome. So here's just, visualizing variants on a linear reference genome. You have your SNPs, you have your deletions, your insertions, inversions, and duplications, right? The thing that all of these things kind of share is they're a location in linear space on the genome, right? Additionally, gene regulation is also very much viewed in this linear fashion where you have these distal regulatory elements that we know interact with promoters and enhancers, and they're some distance away, up to tens of kilobases. And then you have your promoter and activation factors and where POL2 binds and starts transcribing. But generally, most models, when you see these things, are depicted very much in linear space. And that is largely a result of the assays that we're using to characterize both the variation we see in genomes and gene regulation factors that go into the regulation of genes. That's great. This technology and these viewpoints have really allowed us to expand and evolve our understanding of how genomes work. But what is also clear is that genomes aren't organized in a straight line, right? You have each cell in the human genome has about 3 meters of DNA that it's got to compact into a nucleus. And so when you do that, it takes a series of folding events that really get everything nice and compact into that nucleus. And so this creates this hierarchical structure of folding events that actually has real ramifications for gene regulation. So if we look at the very large scale, large speaking, the size of the nucleus, right, we have these chromosomal territories where different chromatin is packed in different compartments of the nucleus. And then even within a chromosome territory, there's an additional packing that occurs where you get these open, active euchromatin compartments that are often associated with high levels of gene transcription. And then you have these silent heterochromatin, or often called B compartments, that are really compact and silent and not transcriptionally active. Interestingly, we get a lot of SNP occurrences that happen in these heterochromatin, these silent chromatin, these non-coding regions. We'll talk about that in a bit. Now, even if you zoom even further in onto these different compartments, we can start seeing how the genome is being organized in terms of clusters of linked function genes. These are often called topologically associating domains. These are genes that are kind of isolated off in their own physical cluster that kind of share share a pathway of different functions, right? That are all kind of linked to each other. And then you can zoom in onto an individual gene and really start asking the question of, what are the transcription factors and enhancers that are interacting with a promoter that actually kicks off gene regulation, right? And so at all of these scales, genome variation can alter how these things are compacted and shifted and viewed and compartmentalized, right? And so a big focus of today's talk is thinking about how that 3D orientation can, one, either help us make sense of those variants, and then the second part is how do large structural variants impact this folding structure. And so the bottom line here is that conformation or 3D genomics, what it really does for us is it helps us understand that transcriptional landscape of chromatin, where we're moving from this linear view where you have your post-translational modifications, your remodeling complexes and transcription factors that are someplace downstream from your core promoter and pull to binding and transcription into this three-dimensional view where we can actually now all link these things in physical space and say, yes, the enhancer marks are interacting with this promoter region, right? And so it really helps us move from this very linear domain into this 3D orientation that's really reflective of how genomes are actually packaged inside of a nucleus. So how do we get there? How do we get from this linear view of transcription into this three-dimensional view of the genome? The answer is chromatin conformation capture, or 3C technology. This has been around for about 20 years-ish, give or take. But it all kind of starts with the same core workflow where you start with cross-linked chromatin, and then that chromatin is digested. Historically, that's been done with restriction enzymes or a mix of restriction enzymes. We're going to talk about how that's probably not the best way to move forward with this assay. But regardless of however you digest that chromatin, the next step is to perform proximity ligation, where these free ends ligate back to themselves, and those ligations are reflective of not what's close in linear space, but what's close in three-dimensional space. And then when you clean up those proteins and reverse those cross-links, what we're left is chimeric DNA. And that chimera event is representative of some physical distance that's being captured in that ligation event. If you think back to mate pair libraries, how they make circles and the size of that circle, and so the tag at either end of that biotin is representative of like 4 kb or 10 kb, it's kind of the same idea, but just kind of on steroids, where we're capturing all of those three-dimensional events through that chimeric molecule generation. Now, once you have this chimeric DNA, there are many things that you can do with it. And the original approach was kind of to take a qPCR-based approach where you have two loci and you want to ask how frequently are they interacting. And so you just hit it with a qPCR probe and ask how many times do I see this chimeric read or chimeric molecule. The next approach was to do 4C, where say you're really interested in all of the particular chimeras that could form with this blue region. That's great. So you make all of your chimeric molecules, and then you convert those into a circle, and then you do inverse PCR, where you're flanked on your anchor that primer to that known site that you're interested in, and then you amplify the region of unknown chimeric binding. And then from there, you could go into sequencing or a microarray approach. The next would be sort of a 5C or hybrid capture approach, where you have several regions that you're interested in. Say that these are represented by a handful of these red regions. And so you design probes that go in and capture or that hybridize to that known region, and then you bring these unknown blue regions down for the ride when you do the pull down, and now you can ask the question, what unknown blue regions are interacting with my known red regions? Then we have Hi-C, which is most commonly used and has been most widely adopted, which is where you biotinylate this chimeric event. You do a streptavidin pull down on that biotin, and then you generate a sequencing library and just do NGS sequencing on that and look at all of the interactions that could be occurring. Then, if you're interested in going even further, you could do a protein-directed approach where you introduce a chromatin immunoprecipitation on an antibody of interest that pulls down on a protein, and so you get a more protein-centric view of what's binding to the chromatin and what interactions are associated with that particular region. But today, we're really going to focus on Hi-C, what its current limitations and capabilities are, and then how we here at Dovetail have really been trying to engineer the best version of that for end users. So now you have your chimeric DNA, it's been biotinylated, you've converted that into a sequence library. What does the data actually look like? Well, it looks like this, where we have the genomic position on your x-axis and your read pair separation on your y-axis. And then you have all of these dots, and each of these dots is reflective of a read pair, where read 1, if we're looking at this dot, maps to this region, read 2 for this dot maps to this region, and then all the way out here, we're looking at much larger events. And these events can span from, single base pairs, because this is just NGS sequencing, you'll capture single bases along this read, but then they also span megabases, where this read pair separation can be as large as basically the chromosome. And so it's a very powerful data type. And then what you can start doing is asking of those loci, how frequently do those two regions interact? And so here we're looking at real data, where if we just say this is chromosome A, this is chromosome B, we can start seeing where there are hotspots. The red indicates the most frequently interacted regions, and you can start seeing how this genome organization of this loci interacts with this a lot more frequently than, say, this region and this region, right? These two are separated from each other. They're isolated in really their own cluster. So it's a very powerful data type that gives us information, again, all the way from that single base pair, because this is whole genome sequencing, right? You're sequencing DNA molecules just like you normally would. But now, instead of that insert size being representative of the that shearing process of making a whole genome sequencing library, it's actually reflective of those chimeric reads. So now that we know what conformation can do for us and how you generate that data and what that data looks like, let's look at some really, I think, are phenomenal case studies here that exemplify some of the uses of these data. So just to give a very quick primer before jumping right into the case studies, we're going to be focusing really, like I mentioned earlier, on two areas: the single nucleotide variation or polymorphisms, and these structural variants. And so when we're thinking about a single nucleotide, right, it's good to keep in mind that many of these SNPs and SNVs occur in non-coding regions, and it's really hard to assign what these variants are doing. And so the term that's kind of popped up around them are these VUSs, VUSs, variants of unknown function, right? And the other thing that's often occurring when people are using SNP and SNV data is that they're putting an assumption around the fact that the area or gene that it's impacting is the nearest neighbor, it's the nearest genomic feature, right? So if you're looking at a SNP that occurs at an enhancer region, the assumption often is to put that to the nearest neighbor gene, your nearest neighbor promoter. And our first case study is going to show you why that's maybe not the best idea. And then when we get into structural variation, little is actually understood about how these structural variants impact chromatin dynamics and what exactly is that structural morphology changing in terms of how genes are being regulated. But what has been noted in the literature is it's quite clear that disruption of chromatin topology in general leads to atypical gene regulation. And so we're going to walk through a case study that kind of links that structural variation to alteration of genome topology and what that means for gene regulation. That really gets you to that phenotype that you're observing maybe through RNA-seq type data. All right. So first case study. This is a really fantastic piece of work. Not at all 2019. I think this is a science manuscript. But really what this group was doing was using the interactome data, this 3D orientation data, to help annotate neuronal disease SNPs that have been previously identified through GWAS. So here they used a combination of ChIP-seq, GWAS data, and what's known as high-ChIP, which is this high-C-like data type, to really ask the questions of what are enhancers and promoters doing and how can we use that information to better understand these disease-associated SNPs. And so what we're showing here are the other data types that they've used. So they're looking at microglia, neurons, OLs, and astrocytes here, and they're using ATAC-seq, ChIP-seq with enhancers and active promoters. And they're basically just scanning the genome for where these things, where these open accessible chromatin occur, where enhancers are located, and where active promoters are located. And then they just overlap and say between these different cell types, in terms of promoters, how many of these features are shared, how many are unique to the different cell types. And then enhancers, they're bringing in additional data types, the psych encode data, and asking how many enhancers do we actually know that overlap with each other in terms of these different cell types, and how many are unique. And so really their next goal here is, can they describe why these differences between these different cell types occur and what that might mean for the phenotype of those particular cell types. So they applied this chromatin immunoprecipitation data type with high C, known as, again, as high ChIP or plaque-seq, to build up this interactome view that's associated with enhancers and promoters. So here's the ATAC-seq signal, here's your enhancer signal, here's your promoter signal, and then here is the interactome data, where we can start using that three-dimensional linkage to say what regions of these genome are actually interacting with each other. And so what they were able to find here is that in microglia, they found this really accessible region of the chromatin that is, almost 500 kilobases downstream from this gene here. And then when they look at the enhancer signal, they see that, again, this open region is associated with this enhancer. And we also see that there is an enhancer that's occurring right here, that's right next to the gene. But what we're also seeing when we go in and look at where the promoter is in reference to the enhancer, so keep in mind there's an enhancer that occurs right over this promoter as well. They see very little interaction between its nearest neighbor enhancer to that actual promoter. But what they do see is at this super enhancer region, where there's over or hyperacetylated regions, that they can link this super enhancer region with a high degree of contacts with this gene, and that moreover in the other cell types, we don't see that particular interaction that's being associated with this super enhancer and this promoter. So this is a cell-specific super enhancer that we can implicate in its interaction with this gene that's, hundreds of kilobases downstream from it, where the, sort of standard thought process here would be that, oh, look, we have an enhancer that sits right over this promoter. That should be the one that's activating it. We actually see very little evidence for that interaction, but what we do see is a high degree of interaction between this distal super enhancer to this promoter region. So that's really great, right? We can now start associating these enhancers with the promoters that they're acting with. So the next step is okay, we know that there are polymorphisms that occur across the genomes in these in neuronal diseases. Can we use this interactome data, this high C data type, to start maybe re-annotating what SNPs are actually impacting what genes? And so what they did here is they've plotted out where these SNPs are occurring, right? And so in this blue bar here, we're seeing that's the SNP that's been annotated or that's been identified through GWAS studies as being associated with Alzheimer's disease, right? And what GWAS has done has assigned this SNP to this promoter of SLC. Right. And so this is the sort of that nearest neighbor approach to, here's my SNP, here's where the promoter is, that's the closest. So these two things are actually linked to each other, right? What we're actually seeing here is that if we look at the interactome data, right, that 3C data type, we can see that this SNP is actually very much linked to these genes over here, the TRIP, the ATXN, and the CPSF, right? And what we actually don't see is any indication that these SNPs here interact with the promoter of SLC, right? And so this is very clearly a misannotation of what this SNP is actually doing. And what we think that it's doing is that the SNP is actually impacting the enhancer location or the ability for these enhancers to occur at this region. And so when we lose that SNP, we lose these connections to these genes downstream, not necessarily the nearest neighbor promoter, right? And so that is one of the really powerful types of information that you can get only through using a high C data type, right? If you were just looking at ChIP-seq, you wouldn't be able to link the SNPs that occur in this enhancer region with the promoters for these genes that are, almost 200 kilobases downstream, right? So that was a really cool finding, and I think highlights some of the usability of integrating GWAS data with this interactome data type, this high C data type. I highly recommend you read this whole manuscript. There's a lot more in there that I'm not covering. This is just a very high-level overview of the utility of bringing in that proximity ligation data, that high-C data into your GWAS studies. And then so moving on into structural variants and really what that's doing to chromatin. This is a really kind of a really cool area where oftentimes people can detect large structural variants, but it's really hard to actually link them into what it is that they're doing. This is a really great manuscript that actually does some really great work incorporating some SNP information there and working at the population level scale. What I found very compelling about this was the structural variation story that they were telling. So here what they're depicting are the types of structural variations that they're observing in reference to the standard GM 12878 human genome, where this is the standard topological linkages, right? And then how those linkages are disrupted when there's a deletion, right? When we delete this B to C region, we lose, everything becomes more compact, we lose some contacts. When there's a duplication, things get elongated and maybe some interactions that you think would be occurring maybe aren't. Inversions, you lose contacts when you switch things around. And then insertions that actually really kind of disrupt the topology landscape quite greatly. So what this group did is they modeled using high-C data what the actual chromatin looping dynamics look like for these various structural variations. And so when you're looking at the normal reference genome, this is what the looping structure looks like if you were to just draw it on a 2D map. And then here's what they're able to show in that three-dimensional orientation, where the colors here in this map are reflective of these regions that are in that 2D pattern. So what you can see here across the board is that when there is any sort of structural variation, being a deletion, duplication, inversion, insertion, that there are dramatic changes in terms of loss of looping events, gain of looping events, sort of reorientating looping events that they kind of look the same, but they're actually flipping various loci in that looping structure. And then how it can really just totally throw off by adding multiple loops into the chromatin. And so I think it's even more apparent when you look at the 3D modeling of the chromatin dynamics from these structural variations, where here this is the normal type, and we can see that things kind of become a lot more compact. You're losing a loop there, you're moving these two regions closer to each other. In this case, you're really distorting that looping shape, and then here it kind of becomes this hodgepodge of various looping structures. But it's all these four are very different than the normal type. And so this group did is wanting to ask, what does that mean? What does that really mean for the regulatory landscape? And so to do that, they took many different cell types that have structural variation and characterized the different structural variants in reference to topological boundaries and topological loops, right? And really what they found is that indels were one of the most common SVs detected in general, and that many SVs occur within a TAD boundary, and that duplications in SNVs are the most common events that occur at a TAD boundary. So really, what they're kind of trying to show is that these structural variations occur across, pretty much all different aspects of that chromatin looping event. But what are kind of maybe things to key in on initially are things that happen at these topological boundaries. So duplications and copy number variations could really, really disrupt this chromatin folding. And so they took an example of that and asked, what actually happens to that structure? So they took the high-C data and they took the inversion and were able to realign the high-C data to that inverted, sorry, it's a deletion, that deleted genotype, and then mapped it out. And so what they saw here is in normal space, right, in your normal situation, this is what the chromatin looks like. And then if you zoom in onto this region, you get a better picture there. But in the deletion SV, it really changes the chromatin dynamic, and particularly we're looking at these regions here that were once very much entwined into that chromatin looping, and now it's way, way, way out physically. The physical distance between this loop where it is now and where it used to be is quite dramatic. And so the next real question is what does that do to the transcriptional landscape? So then they mapped out the introns, exons, and enhancers that are associated with this region. And what we can see here is that particularly this PP2R gene here is now very, very much removed from the enhancers, whereas in the normal cell type, it's right next door to this enhancer region. So these structural variations really do impact how chromatin is actually orientated, right? And that the whole idea of chromatin orientation impacting gene regulation plays in very nicely here, because if your SVs are disrupting that chromatin dynamic, you're therefore interrupting the transcriptional landscape that can be occurring, or that's physically present, and therefore will probably change the phenotype that you're going to observe in the RNA-seq data. So just a few take-homes. I kind of dropped these in as we were going through, but for the first case study, topological interactions, that interactome data type can really help us assign these variants to their gene of function, and that nearest neighbor approach might not actually be that informative, right? To assigning what gene is being impacted by what variant. And really that confirmation signal, that interactome data, can be helpful in reassigning SNPs in GWAS data. Now, if you think about this from gene or that SNPs that occur in non-coding regions, the Hi-C data type now gives you the power to actually link what is interacting with that SNP in that non-coding region. So that's where we think this data is really powerful in terms of thinking about GWAS data or SNV data. Again, for structural variation, that physically, these structural variants alter the chromatin dynamics, and that has downstream consequences for that promoter enhancer landscape that really does impact transcription, right? And so it's kind of linking those two pieces of information together that can really give us this view of how these structural variants can actually lead to a phenotype that you can observe in the RNA-seq, right? It's all about getting to that mechanism of action rather than just detecting a phenotype, right? That's really the punchline here, is that Hi-C data, that interactome data type, really allows you to more fully and completely describe the mechanism of action that arises to the phenotype that's being observed in the RNA-seq data. Okay, so now let's talk about integrating this interactome, this 3C data, into what you might already be doing in whole genome sequence-based approaches. So let's talk about the barriers, right? Because they're real, they exist in integrating as many data types as humanly possible into these studies, right? One of the biggest barriers is having multiple data types to address these approaches. You need whole genome sequencing panel or array type data to really classify your SNPs and SNVs. Then, you might need long-range reads like a PacBio and Oxford Nanopore, or do optical mapping to really kind of suss out these larger structural variations, right? Having that long-range information is really critical in able to characterizing structural variants. And then if you wanted to integrate that 3D interactome data, that's another library and another sequencing run that you have to do on top of that, right? And so that brings me to my next point, which is sequencing cost, right? Sequencing cost, the cost per base has gotten cheaper. There's no doubt about that, right? Hi-seq, Novo-seq have really kind of been instrumental in bringing that sequence cost down. But if you're trying to do this on a population scale, or if you're really trying to dig in deep on the mechanism of action, that's a lot of dollars that you're putting towards sequencing. And which has really kind of forced people to go into these hybrid capture panels and arrays like exome sequencing and things like that. And when you do that, you reduce your sequencing costs and you increase your resolution over those features, but you're also putting on your blinders to what else is going on. And so what we're going to talk about next is how our Omni-C data type really integrates all of those different approaches in a single assay. So in a single sequencing run, you'll be able to capture all of these features, right? And so just to kind of orientate you to what Omni-C is it's Hi-C at its core. We're not greatly altering that proximity ligation workflow, that core workflow that I outlined at the beginning, with cross-linked cells, fragmentation, proximity ligation, cross-link reversal, library generation, and sequencing. It's the same core workflow. But what we do is we don't use restriction enzymes. Here for our Omni-C protocol, we use DNase. And what DNase does for us is it gives us shotgun-like or whole genome-like coverage of the genome. So the reason why Hi-C data historically has not been integrated into the SNP detection world and into the variation world as much is simply the sequencing coverage is pretty bad. So here what we're plotting is the coverage in terms of X and number of bases that exhibit X coverage. And we're plotting this out for a single restriction enzyme Hi-C, multiple restriction enzyme Hi-C, and then our Omni-C approach. The dotted line here is just whole genome sequencing data, shotgun data, where you just DNA extract, fragment, add your adapter, size, select for the right fragment size, and then you drop that on your sequencer, right? And so you get that nice normal distribution. It's nice and tight, right? So if you wanted to move the mean of this coverage over, it's 10x sequencing more would shift this peak over 10x for the most part. Now, if we compare that to restriction enzyme-based data, there's a lot of regions of the genome that exhibit no to low coverage. And on the other side, there's a big portion of the genome that you're just way over sequencing. So if you wanted to move this mean over, you're going to sequence over here a lot more than you need to, and you're really never going to bring this part of the genome along for the ride. So it's a really inefficient sequencing strategy. Now, if you add more restriction enzymes, it helps, right? It kind of normalizes the curve a little bit better. It gets rid of this sort of bimodal histogram, but you still have this region here that are poorly covered, and you still have these regions here that are overrepresented. And that comes from your enriching over restriction sites. When you biotinylate that restriction site and pull down, all of your data is going to be restriction enzyme focused, right? And so regions that are restriction enzyme deserts don't have any data because you have nothing to biotinylate. Regions that are chock full of restriction sites are over biotinylated and therefore over sequenced. When you move away from that, we're able to get closer to that normal distribution where moving your coverage over to get the coverage required to do certain analyses, you just have to sequence a little bit more to move that curve, right? And it moves very uniformly, unlike with using restriction enzymes. And to hammer this home a little bit, if you just plot these data out on an IGV, right, you can see that in restriction enzyme-based data, there are just these gaps where you have no read pileup, right? And that's associated with those lack of restriction sites. Whereas with our DNase approach, we are getting very uniform data, and even regions that might be harder to sequence, we really don't see these exaggerated gaps that almost go all the way back to the single base level. And so what that does for us, what that genomic coverage really does for us, is it makes us be single-end shotgun data, whole genome sequencing data, or paired-end long-range data, which opens up this whole new world of applications for the use of Hi-C. So from a single library, you can detect these large structural variations through these contact matrices where you can see clearly that this region of chromosome 10 is interacting with this region of chromosome 13. That's a translocation event. Boom, it's right there in the data, right? SNP and indel genotyping you can do because we're shotgun data. We're whole genome sequencing data at its core. You can do genome assembly, which is a hallmark of using Hi-C data because you can now piece together your scaffold, or you can piece together your contigs based on that long-range information. You can now haplotype phase. We'll get into all of this in a bit more detail here in the next few slides, but that the long-range information can build this really nice big haplotype blocks. And then also now you can start incorporating chromatin conformation in your whole genome sequencing studies just by doing one assay. So we'll talk initially about this de novo scaffolding method, which is very near and dear to us at Dovetail. We started as a genome assembly company really using that long-range information to piece together these scaffolds over megabases, right? And because our distance, the physical distance that our reads span, are really limited only by the length of the chromosome, we actually kind of consider PacBio and Oxford Nanopore as short-range reads. You're talking about kilobases. The high-C data type, you're speaking in terms of megabases, right? So that really allows you to piece together these scaffolds into these, or the contigs into these really nice long scaffolds that are almost the size of the chromosome. Now, what this means for you all, if you're thinking about, genome variation, is one of the more popular ways to detect variants is by doing genome assembly and then comparing two assemblies where you have your reference genome 12878 and then you take your cancer HCC 1187 and then you assemble the HCC 1187 and say, where are there differences? Where are there physical distances between these two differences in between these two genomes? So that is a very ground up approach to actually assessing genomic variation, right? It is through this genome assembly approach. SNP detection. So here we're showing shotgun data compared to Omni-C and restriction enzyme high-C. We're getting very good sensitivity and precision. This is something that we're always working on and trying to make better. But through that, that ability to identify SNPs, right, we can now take these variants and phase them and build very large phase blocks. So you're looking at a 250 megabase phase block with Omni-C high-C data, and compared to shotgun data where you're getting maybe like a 28 KB, 30 KB phase block. But the thing to note here is that, yes, the shotgun data gives you a lower switch error rate. Omni-C is about 1%, but if you were to use a restriction enzyme-based approach to phase, your error goes up to like over 3.5%. And that's because Omni-C catches more SNPs because we're not piling reads over restriction fragments. We're just piling the reads over the genome fairly uniformly. So we can detect those heterozygous SNPs, and then with that long-range information, we can build these nice long phase blocks. Then, right, what we can do is once you have your genome, or if you're aligning your data to your reference genome, you can look at the structural variation. And so what I'm showing here is the HCC 1187 BL line that doesn't really have structural variation, and we're comparing that to contact matrices from the cancer line HCC 1187. And here we're looking at that contact matrix where at any given point you say, okay, here is the interaction frequency between any two given regions of the genome. And here we're pointing out a known inversion in the data, and we can say, yes, here are regions of the genome that are interacting, and that signal is way far off the axis. So you're not, that's not normal. That's an inversion event. Here we see a deletion where, again, this is a known characterized deletion in HCC 1187, which isn't observed in the BL cell line. And again, we can see that this signal looks way off axis and it has a nice bright signal compared to the background, suggesting that this is a deletion event between these two regions. And then here's that translocation event that I pointed out earlier, where we can clearly see this region of chromosome 10 interacting with this region of chromosome 13. That's a translocation event. Again, that's not apparent in the BL line. And then the nice thing about this is you could zoom in on these SVs and start looking at what the gene landscape is around these regions. Or conversely, like I said earlier, you could reassemble and look at the topology de novo for these cell lines, right? And then do a genome to genome comparison to a normal. So, all right, that is another powerful data type here where we're allowing you to capture those single nucleotide polymorphisms, and we're allowing you to capture these large structural variants. One of the other very nice things about this approach is it's really quite flexible. Here we're showing just the library quality of the high-C data type, or the Omni-C data type. When we go down all the way to 1,000 cells, we do have low input protocols set up for Omni-C. So if you're interested in lower input, you should definitely reach out to us. We can provide you guidance here. And then the next thing I really want to get into is that second hurdle, which is that sequencing cost. Well, the very nice thing is because we're essentially shotgun data where the physical insert is proximity ligation, that we're compatible with off-the-shelf hybrid capture probes. You don't have to redesign anything to use the probes that you're already using today to maybe describe a cancer panel or to go into an enhancer promoter type panel, right? We're compatible. And so just to kind of demonstrate this, we did this with our Omni-C data type, where we just took a sample, generated the Omni-C library. We split that, and half of it went to normal sequencing to generate the full contact matrix. And then we took a subset of that and did the IDT XGen pan-cancer panel, and then sequenced, aligned both data, filtered bands over those target sites. And then really what I'll show you here is we're really asking, are we capturing topology and are we actually enriching over those target sites? So here is just a snapshot from a genome viewer where we're looking at RPL5, and here is the RPL5 gene. Here are the capture targets here in blue. And then here we're looking at Omni-C coverage sequence at only 1x coverage versus the Omni-C hybrid capture approach only sequenced at 0.3x coverage. As you can see, with 1x coverage is fairly sparse. We expect that at 1x coverage, you're not really going to do a great job of covering the genome. But when we introduce the hybrid capture panel, we're enriching over those target sites, just like you would expect with a shotgun library set or a whole genome sequencing library set. So what we're trying to show here is you don't have to buy new probes. You can use the probes that you're already using. And then what you can do is not only enrich the sequence information over that, you can now enrich the topology signal over that. And so here what we're showing is that contact matrix over RPL5. And what we can see here is that here's where that gene lives. Here is the domain that it sits in. Well, we can see here that from that gene of interest, we can start looking at the contacts associated with that. And we do see this nice cluster here with this smaller topology boundary and contacts that occur within this larger one. And we see some more distal elements that are reflective of contacts that are out here, right? So we're able to recapitulate that topology signal with very, very, very little sequencing by targeting on those regions of interest. So that's where we really think the Omni-C approach is flexible and integratable to what folks are already doing. It allows you to expand your research into doing multiple different applications with a single assay, and it provides you a means to dial in your question with hybrid capture probes that you're already using today to maybe ask your questions, to add that interactome data type to your research to help you maybe re-annotate SNPs in a little bit more of a concrete manner where you have physical linkages that you can concretely point to, or to say, these are how these structural variations are actually impacting the chromatin state and structure, which would then arise to the phenotype that we observe here where a gene gets turned off. Well, it gets turned off because that promoter has been moved so far away from that its target enhancer. So with that, I would really, again, like to say thank you for listening in.